We have Professor Sukhdev Roy, and he will be speaking on optical biomolecular computing with photochromatic proteins. Professor Roy is reader in Department of Physics in the Albag Educational Institute. He obtained his bachelor's and master's degree in physics from DEI and PhD in physics from IIT Delhi. He is a very active researcher and has widely traveled to various educational and research institutions all over the world. He has several publications in refereed journals and conferences. His research interests are biomolecular computing and all-optical switching.
Thank you, Professor Adhar. Well, the title of my presentation is Optical Biomolecular Computing with Photochromic Proteins. Now, what I would do is I present a motivation for studying this, the objectives which we have framed over time, and then I'll focus on all optical switching in photochromic proteins and how that can be applied to all optical biomolecular computing, and then how do these proteins or the response compare with other organic molecules, and then finally make a conclusion. Now, you see, information is what we are all dealing with, and the fact is that the demand for information is exponentially increasing, and therefore the technology which can meet these requirements of high bandwidth, high speed, good reliability, small size and low cost appears to be photonics, which has made an impact on these four key areas of information processing. So you can acquire, transmit, process and store information all in the optical domain through sensors. Optical fibers are the backbone of communication technology through integrated optic circuits and optical disks and holographic memories. Now, if one looks at these two aspects of information processing, that is computing and communication, we find that there is intense effort on studying optical computing. So can you use light for computation? So you can use bulk materials and then modulate or switch light with light. Or probably look into the DNA molecule, which is the building block of life, and do DNA computing. Or find two stable states of a molecule such that you can implement digital or binary logic. So it could be chirality, or it could be change in their electronic states or structure or shape, etc. Or the exotic quantum computing. So the fact is, in all these, you find that the implementation in the optical domain is very attractive. So whether it's molecular, quantum or optical. And the question is, can you have a hybrid approach? So that means, can I find a molecule which is photosensitive and I can manipulate its states with light and therefore effect optical computation or optical molecular computation? Even if one looks at quantum computing, optical quantum computing has several advantages. And another interesting question is, can there be a common platform for integrating technologies? So can one think about a common platform which can be used for molecular, nano or quantum or optical computing? Now, optical computing has been there for a while, and there are several advantages. Of course, high speed, high bandwidth, high interconnection densities, low crosstalk, inherent parallelism, higher fan-in and fan-out capability, immunity to EMI, etc. And the challenge is, can one do most of the functions with light itself? So can one have the magical capability of all optical computing? If one looks at communication, you find that fiber optic communication is of course going to stay there, but the fact is that the power which is offered by fiber optics is not exploited to the maximum, and that is because there is an interconversion which takes place between the transmitter and the receiver end, between electronics and optics, and therefore this limits both the speed and the bandwidth of the system. So the fact is, can one again replace the devices of processing, right, instead of electronic or optoelectronic to all optical, right, devices or information processing? So the present challenge seems to be that one would have to look into how one can achieve all optical information processing. Now, in order to have all optical, that means everything optical, then one is looking at acquiring absolute optical control over all these basic features of information processing. So that means one has to control light with light, and therefore it is necessary that one looks into the principles of nonlinear optics. Nonlinear optics is a very well established branch of physics, but the problem has been that in terms of practical limitations, that is practical materials, right? So we need materials which have high nonlinearity so that at very low powers you can observe nonlinear effects. They should have a fast response, high efficiency, stability because it's a photo material, and therefore, and finally it has to be a cost-effective proposition. Now, several materials have been looked into. They are still being researched. So you have liquid crystals, semiconductor doped glasses, organometallics, organic dyes and polymers, fullerenes, etc. But an interesting development is the advent of biomaterials. So this is very interesting. That is, a new class of materials probably could help us in achieving all optical information processing. And of course, there's a fundamental need to understand how information processing takes place in nature as such. And probably it could lead to a more cost-effective, environmentally safe technology, and of course the application of biotechnological techniques might be used for tailoring the response. And of course, simultaneously we have the power of nanotechnology to synthesize, characterize and design novel molecular configurations so that one can tailor the molecular nonlinear optical response to achieve these capabilities. Now, this means that we are looking at a very interesting proposition, and that is the integration of the three thrust areas of modern science, that is integrating nanotechnology and biotechnology if one is looking at biological materials for information processing, and that is a very niche area of nanoBiophotonics. Now, let us try to look at what happens on Earth. So light sustains life on Earth, and if you look at what happens around us, everything has adapted to ambient light levels. So whether it is in the sea or oceans, or whether it is on the surface of the earth. So an interesting feature is that you have plants which have a tendency to grow towards light, you see. So what is it that triggers this response? So this is a very high-tech area of interest in molecular biology. So molecular biologists have been able to isolate these ultra-sensitive photoreceptor proteins which essentially dictate this response. Similarly, in the mornings you have these beautiful flowers which flower, and in the evening they close. So there is some molecule or sensor which is taking track or keeping track of circadian timing. So these are very, very interesting things which we see all around us. And if you look at the ocean masses, you have in the depths of the ocean where light intensity levels are very small or negligible, you have diversity of life which survives. So the fact is, how do these organisms, exotic organisms, survive in such ambient light conditions? Now, we find that these are due to natural photoreceptors, and they are abundantly found throughout the plant and animal kingdom. They've been optimized over centuries of evolution. They have a unique photoreaction behavior. It's a very rapid and ultra-fast response. They have a very low light intensity response. You can purify these samples. You have the flexibility of modifying their properties by physical, chemical and genetic engineering techniques, and of course they can be synthesized without the use of any hazardous chemicals. Now, if one tries to think about a biological material which is very sensitive, well, you think about the human retina. So this is what makes us survive every day. So the human retina is very sensitive, human or any retina, visual retina. But the fact is that the light activating molecule, which essentially does the processing, is a rhodopsin protein. So it is there at the retina, and the fact is you can't have that for device applications. But interestingly, you find that this rhodopsin is a big joint family. So you can classify these two into two categories. One is the visual rhodopsins. These are photosensitive pigments which are found in eyes of insects or animals or humans. And there is another class of rhodopsins which are called apile rhodopsins. Now these are light-driven pumps, and several of them have been discovered over the years. Bacterial rhodopsin, which was discovered in 1971, halorhodopsin, photosensitive rhodopsin, eukaryotic rhodopsins, and a recent discovery, proteorhodopsin in 2000. And you find that through bioinformatics, if you map the sequence of this protein, the rhodopsin protein, you find that it is almost all pervasive. So you find it prevalent in salt flats, soils, fresh waters, fungi or archaea, proteobacteria, etc. So nearly 800 relatives life-sustaining activity of photosynthesis. So therefore, you can imagine how far their photoresponse has adapted over so many changes on our planet. Now, one of the most important and most interesting material, outstanding photonic material which has been discovered is bacteriorhodopsin. So this is grown by a bacteria, so therefore it's called bacteriorhodopsin. And this is found in the purple membrane in saltwater marshes. And if you synthesize these membranes, you find it contains the same rhodopsin protein similar to the visual rhodopsin, which you find in the human retina. Now, let us look at the photoresponse of this interesting molecule. So this molecule in its native state is very sensitive to green-yellow light, although it absorbs the entire visible spectrum, but it is very sensitive. It has a peak absorption wavelength of 570 nanometers. So if you hit it or excite it with a photon, it undergoes conformational transitions. So conformational transitions means that the shape of the protein changes, and it undergoes these conformational transitions and spends some amount of time in each transition, in each state. And these subscripts denote the peak absorption wavelength of these states, and these are transitions which take place in some amount of time. So this is very fast, sub-picoseconds. Then in 3 picoseconds it goes to another K state, the L state, and finally it goes back and relaxes to the initial state. Now, under certain situations or under certain genetic mutation conditions, you find a variant in which if you shine a red light, it goes to a state where it is stable for infinite amount of time. So these are the P or the Q states which have a peak absorption at 490 and 380 nanometers. So the molecule does not come out of these states, and it's very, very stable for infinite time. But the interesting thing is that it is sensitive. There's no thermal relaxation here like the other states, but you can induce it to come back to the initial state if you shine blue light. So you can shine blue light and make it go back to the initial state. Another interesting feature is that at any instant of time, if you shine another wavelength, you can truncate the photocycle and you can make it switch back to the initial state. So you can see that the entire visible region is mapped into its different states, and these lifetimes, the spectral characteristics can be changed through various techniques. This is the absorption spectrum, and you can see this is the absorption spectra of the BR's native state, and this is the Q or the M state. So there's a large change in the absorption, and that means that it is a photochromic material. So it's a photochromic material, reversible transitions, and you can make it go back to any of these states by shining appropriate light or leaving it for a certain amount of time. This is what shows you the change in the conformation when it absorbs a photon. So this is the old transretinal and this is the 13 cis retinal. So there's a change in the shape around the bond which has occurred after it absorbs a photon. Now, in the native state, just the molecule acts like an AND gate. So when both the inputs are 1, it goes to a higher excited state. So you can have a 0 here or a 1 labeled for the P or the Q state, and you can have a simple AND gate operation using this molecular photocycle. Similarly, here is a comparison between the wild type and a mutant D96N, and what has to be appreciated is the fact that all these higher excited states are absent in this photocycle. So you can just tailor with the photocycle depending upon how you use genetic engineering. Now, there are very unique properties and advantages of bacteriorhodopsin. It has a very efficient photochemistry, high absorption cross-section, high quantum efficiency. It absorbs 65% of light which is incident on it. It has a multifunctional response, photochromic, photoelectric, and a proton pump. It has a large change in the refractive index, large shift in the absorption spectra. You have transitions which are reversible in the photocycle. The most important factor for a photosensitive material is photostability. So how stable is it? The interesting thing is that it is stable over a very high temperature range. So even at a temperature of 140 degrees centigrade, it doesn't degenerate or degrade. And it has a stability over a wide pH range from 0 to 12. And even after a million microarray cycles, the material does not show any photodegradation. So this just surpasses any other organic material reported today. And of course, you can tailor its properties and its environmental friendly. These are some of the optical properties of BR films. The interesting thing is that this is just one protein. There are several others which have been discovered, and one of them which I have shown here is Rhodopsin. You can see that it also has a photocycle. This is the absorption spectra. Interesting thing is that it is sensitive to blue light. So a blue sensitive material is of tremendous importance in optical memories. And similarly, you have this Rhodopsin, which is sensitive to 520 nanometers, and it has a longer lifetime photocycle spanning about 153 milliseconds, important for memory applications. Similarly, there's another interesting system, and that is photoactive yellow proteins. This again is a blue sensitive material, and it shows a photocycle similar to what you have in the other proteins, but there are very subtle differences here, and one of them being that the excited state absorption cross section at the peak wavelength corresponding to the PR state or the I0 state has a higher value than the ground state. So this is satisfying a condition which is called reverse saturable absorption, which is very important for switching applications. Similarly, if you look at sensory photoreceptors in plants, you have three main families: phytochromes, phototropins, and cryptochromes, which are sensitive to different regions of the wavelength of these visible spectrum. So these are blue light sensitive proteins, these two, and you have the red light photosensitive protein, which are phytochromes, and they govern different kinds of responses in plants, right from stomatal opening, circadian entrainment, flowering, germination, detiolation, phototropism, that is the tendency of the plant or flower to grow towards light, etc. So this is another photocycle of a protein isolated from oats, and you see that it has a simple photocycle with just 3 states, 1, 2 and 3, and there's a large shift in the spectrum. So from 450 you go to 660, so there's a very large shift in the spectrum, and therefore this again is a photochromic material with a slightly longer lifetime of the last state. That's the 70-second lifetime for the transition. Another interesting material which we all are aware of is chlorophyll. Now chlorophyll A, B, C, D, there are different kinds of chlorophylls which have been identified and studied over so many years. And this is the energy level diagram which can be used to explain the electronic energy transitions for chlorophyll A. This is the absorption spectrum. Another interesting fact here is that there's a broad range of spectrum, the wavelength range, where the triplet state, that's the triplet state, has an excited state with absorption cross section which is more than the ground state absorption cross section. Now, interesting proposition is, can these molecules be used for information processing? Now, there have been several applications which have been proposed with BR. So Rhodopsin is a prototype. It's a model system which is being studied by molecular biologists and even for information processing. So these different properties of this molecule has been used to propose different kinds of applications. Very important ones like ATP generation in reactors, desalination of seawater, conversion of sunlight to electricity, artificial retina, right? Of the photochromic properties have been used mostly for information processing, right from pattern recognition, bistability, holographic memories, optical displays, neural networks, etc. And of course, biosensors and radiation detections are other kinds of applications which have been. This is a schematic of an optoelectronic ICE which with a photodeceiver, biophotodeceiver that is constructed out of Rhodopsin. This is a schematic of a 3-dimensional memory which has been proposed and has been demonstrated with Rhodopsin, which has been doped in a polymer gel in a cuvette. So like I showed the photocycle there, if you remember, a green light and a red light can act as a write beam. So you can write or make the molecule go to the P state, and if you shine a red light, then you find that all those states or the molecules which are in the excited state, they do not absorb this red light. So you can just map where exactly the absorption or transmission is high. So you can read data, and finally you can erase data if you shine blue light, and therefore this is the prototype of a volumetric optical memory with Rhodopsin, and this is a prototype which has been shown and demonstrated to work along with the traditional computer. So a 3D memory would enhance the storage capability to 3 orders of magnitude. Now, the basic building block of information processing system is a switch, and the fact is, how can one design a switch, and that too an all-optical switch? So our objectives have been to look into the nonlinear optical response of these photochromic proteins and see how one can design an efficient switch and then look into the applications for all optical computing. How can one optimize the photo response, right, to achieve the desired designs, and then can one achieve low power all optical biomolecular computing devices, and would there be advantages over other organic materials? Now, there have been several designs which have been proposed in the literature based on optical bistability, dynamic holographic gratings or degenerate four-way mixing, photo-induced dichroism, integrated optic configuration, self-diffraction. But we focused on a very interesting, a very simple mechanism, and that is excited state absorption. So we look into this basic configuration. So you have a protein film or a protein sample. So you excite this with a pump pulse laser beam, and the excited molecules are then probed by a CW probe laser beam. So you have therefore the molecules which get excited, the photocycles are activated, and in the excited state they conform to a state which has an absorption of this particular wavelength. So therefore you have a mechanism by means of which you can control light with light. So with the pump probe technique, one can, in a very simple manner, without touching, without biasing or putting any external bias, one can just use the photochromic property of the material to induce optical switching, and therefore look into some device applications. So if I have a probe beam, it passes through this sample because this probe beam is not absorbed by the ground state. But now if I pump this with a green light, then I find that the excited molecules absorb this probe, and therefore I can modulate this probe beam. So it could be a CW implementation, continuous wave, or it could be a pulsed implementation. So here I have a pulse which excites the sample, and therefore the excited molecules absorb this laser beam at this particular wavelength, and therefore there is a switching operation. So the first thing is to have an accurate model of this dynamic. So the fact is, if one looks into the steady state analysis, one can take into account all the transitions which are there reported in the literature in the photocycle, and one can therefore write down the expression, a general, exact and analytical expression of the absorption coefficient, right? It's a nonlinear expression in terms of the absorption cross sections, the rate constants, right, lifetimes, quantum efficiencies, etc. And one can make it into a very compact form in terms of the powers of the intensities of the modulating beam. So with this, one can therefore accurately look into the theory as well as the experiment. So here these coefficients A, B, C and A prime, B prime, C primes can be written in terms of all the constants and parameters which are required to characterize the photocycle. And therefore, one can look into the transmission of the normalized transmission of different probe wavelengths, right, at different wavelengths, and see, depending on the spectral characteristics, what kind of states would be activated, excited, and which states would absorb that probe beam. So this is an experiment and a theory. This is for PPR, Ferronis photorhodopsin, and you can see that different wavelengths have different response depending upon the pump power. So as you increase the pump power, you can modulate different wavelengths in a different manner. Similarly here, you have this mutant, and you can see that in microwatts per centimeter square, you can have very high modulation depths as high as 50% for a concentration of 1.6 into 10 to the power minus 2 mole per liter. Here is a result which shows you the variation for different mutants. So you can see that at different pH values, you can tailor the lifetime of the excited states so that you have totally contrasting transmission characteristics. So if I have a characteristic like this, I can implement an XOR logic gate by using two intensity laser beams at 498 nanometers by considering a suitable threshold. So that would be a very low power implementation. Similarly here, for another state, that's the KL state, I have a 512 nanometer light passing through, and it is controlled by a blue light, right? And one can see that the transmission goes very high, that high as 96.68%, and you find that there is an optimum value of a normalized coefficient beta, which depends upon the concentration, the cross section, and the length of the sample. So we've done rigorous analysis, and depending upon the transmission characteristics, we propose that using different threshold values and multiple pump intensities, one can design different kinds of logic gates in parallel. So if I have a film, I can implement an OR, AND, XNOR, NOR and NAND gate by using laser beams at different parts of the film. So here we've used two different films with different parameters or different characteristics to implement using a common threshold, but one can use the same film but using different thresholds, one can implement parallel logic gates with continuous beams. Here is the schematic again of the pulse of the dynamic case. So you have a pulse response here. So this one can model by solving the nonlinear coupled differential equations using a Gaussian beam, pulse beam for the pulse laser. And these are the results here. So you can see that the transmission for a blue beam, which is controlled by a 570 nanometer pulse, having different kinds of characteristics depending upon the lifetime of the M state. So one can have either a peak or a dip, and this can be explained on the basis of the population dynamics and the effect of different parameters like pulse width, intensity, etc. One can tailor the response. These are experimental curves for nanosecond excitations in PPR. So you can see as the peak intensity increases, more number of molecules get excited, and therefore the dip in the transmission increases. Similarly, this is at a different wavelength. You have a higher peak coming in here because of less absorption. These are again very interesting results, and these results show that if I have a probe intensity which is very strong, this is the case when you have a weak probe, but as you increase the probe intensity, you find that there's a total phase change in the switching characteristics. So I can flip the switch, right, from a peak to a dip by just controlling the intensity of the probe beam. So if I have two pulses, or if I have multiple pulses for the pump beam corresponding to that wavelength, and I have a continuous laser beam or probe here, I can effect a 2-bit logic gate. So this is a simple switching characteristic. So a pulse comes in and you have a switch here, so this is a NOT gate. And if I have two pulse strains coming in the input, then each pulse gives me an increase in the intensity if the lifetime of the excited state is small, so that there are no molecules in the excited state. So I have a peak coming in, and if I have two pulses coming in phase together, then I have a higher peak coming in. So therefore, using a suitable threshold or not, I have an implementation of an AND gate or an OR gate. Similarly, if I have a film which has a higher lifetime of the excited state, then I would have a dip, and therefore this dip would be more because of the intensity of the pump intensity, because of the two pulses coming in phase. So therefore again, if I do not use a threshold, or if I use a threshold appropriately here, I have an implementation of the universal NOR and NAND logic gates using a protein film. This is the effect of the pulse strain, and you can see as the pulses come nearer, the molecules are not able to relax back to the original states, and so therefore the modulation decreases and finally saturates after a certain value. These are the comparison between experiment and theory again for the O state dynamics. So you have a red light from a helium neon laser which is acting as a probe, and you have a 532 Nd:YAG laser beam which acts as a pump. So these are the switching characteristics, experimental, and we've modeled them with a super Gaussian pulse to approximate these pulses to a chopper, and you can see that the results are in very good agreement with experiment. So these experiments we had performed way back at TIFR. This is the experimental setup for a nanosecond optical switching in another protein, that blue sensitive protein. So you have an Nd:YAG laser through a chopper, excites the sample, and through a monochromata you have different wavelengths passing through this sample, and they are detected by a photomultiplier tube, and you have the results on the oscilloscope. So these are the results which you see, experiment, and you see what is the effect of the increase in the frequency of these pulses. So if the pulses are separated apart, so the time the pulse width is large, you have a perfect rectangular switching characteristic. But as you increase the frequency, you find that the shape of the pulses changes, and there's an apparent phase shift between the pump and the probe. So it's a very interesting thing, but after a certain while you find that you have a noisy characteristic. But interesting thing is that we modeled the response through the theory, and you find that the theoretical characteristics or results are in very good agreement with the experimentation. the experimental results. Another interesting fact is that if you pass different wavelengths or different colors of light, you find that the pulse shape can be controlled, or you can have a pulse shape. So you have a triangular or a sinusoidal, right, pulse shape at different wavelengths. Again, the theory and the experiment are in very good agreement. These are the results, switching results for proteorhodopsin, and this is for the plant love2 phototropin, and you can see that the switching time is of the order of 1.6 microseconds and 22.3 microseconds, and you have a very large percentage modulation, about 30%, 31% here. So changing the concentration of the sample can lead you to a different, a high modulation rate. These are the theoretical results for switching in chlorophyll A sample from spinach leaves, and you find that a combination of different wavelengths give you switching contrast as high as 96. So if I use 442 and 476, I have a 96%, and the highest is for a red light and a blue light, that is as high as 97.5%. So again, the switching time is of the order of microseconds. These are the experiment and theory results, again for chlorophyll, and which are again in very good agreement. These are the switching characteristics for photoactive yellow protein, and we find that this is much faster switching, and this takes place in sub-microseconds, nanosecond switching. So if you compare these different photochromic proteins response with organic materials, which again we've researched ourselves. So you have the platinum ethanyl complex, C16 toluene or polymethine dye, right? You find that you can have a high contrast or a low contrast depending upon the pump intensity. The switching time goes from milliseconds to sub-microseconds, and you have very high switching times here for organic molecules in nanoseconds, but with gigawatts per centimeter square. So there's a trade-off between the high contrast and low power operation, and of course fast operation with high contrast. So there are certain factors which need to be taken into account, and most important, the absorption cross sections at different wavelengths. Now another interesting development which has taken place, and which is a very interesting field of research these days, is the advent of microresonators. Now these microresonators have been proposed in different geometries with different materials ranging from silicon to silica, and they have extreme advantages. They're very sensitive, low switching threshold, high quality factor as high as 10 to the power 9, ultra compactness, on-chip integration, and you can tune the properties by changing the shape of the resonator. Now, let us look at the principle of operation. So the principle is the following. So you have a laser diode, and you pass light in an optical fiber or a waveguide in integrated optic geometry. You have a detector here, and you have a microsphere, a silica microsphere in this case, which is just touching the surface of this optical fiber, and in this case it's a taper. So if I form a tapered fiber and I place a microsphere on top here, something very interesting happens, and that is that if I scan different wavelengths from the laser diode, one particular wavelength would couple along the surface or the circumference of this microsphere. So that would satisfy the modal condition, that is the condition for whispering gallery modes. And therefore, in the transmission spectrum, if I scan the wavelengths in the transmission spectrum, I would find a dip at those wavelengths which satisfy the whispering gallery mode condition. So I can measure this wavelength, and I can therefore try to detect what is happening or what is happening. It's very sensitive to the resonance condition, so it gives me a flexibility of trying to detect what is happening at the surface of the sphere. So the interesting thing is that a single molecule, if it attaches to the surface, can alter the resonance condition, and in this case the shift in the wavelength can be monitored, and therefore which can be monitored with very great resolution, right, sub-nanometer, and therefore I can in principle detect a single molecule in this resonator geometry. So this is a very interesting proposition, and a group at Harvard did something more to it, and that is they took two optical fibers and attached a silica sphere here in between and coated bacterial rhodopsin on it. Now, a very interesting thing, this is a very interesting configuration. Now what happens is, you pass, so there's a wavelength which gets coupled, right, and just couples from this fiber through the sphere to the other fiber. So there's a switch here. But the interesting thing is it's an all-optical switch. So if I shine green light now, the bacterial rhodopsin protein now undergoes the photocycle, right? So there's a conformational change, and therefore the coupling condition or the resonance condition changes, and so therefore there is a different wavelength now which switches off. Not the first one, but a different wavelength. So there's a dip at a different wavelength, right, on the activation of this protein. So these are the photographs. So you have light which passes through here, and as soon as you activate, it just goes and gets coupled here. So this is an interesting proposition to look into all-optical switching, right, with the advantage that it's a very low switch, and using the advantages of microresonators with high quality factor, there is absolute switching of the intensity. So there's a very low loss which that takes place. So at output port 2, there's a very small amount of power which just goes in. So 96% light just couples here. So that means you can have high fan out and cascadability in this system. So collaborating with this group at Harvard, what we proposed is that if you, in addition to 532, if you shine blue light, then you would truncate the photocycle at the end state, and therefore there would be a faster switch. And the fact is that this is the result which we got. So with the input beam and the two input beams at 532 and 405 nanometers, you find it's a much faster switch, which is about 50 microseconds switch on, switch off time. So a theoretical analysis can be performed, and we've found out the shift in fractional wavelength shift depending on the surface density of the bacterial rhodopsin layer, which essentially depends linearly on the diameter of the microsphere. So this switch, therefore, can be a very interesting way of designing an all-optical reversible conservative Fredkin gate. So what you have here is the fact that you have the switch, you have a BR, so this is the control signal which just passes through, and you have two inputs, one from here and the other, the port B from here, and depending upon the consequences that is decided by the control signal intensity, right, one has output at port A or output at port B. So if you look at the various combinations of A, B in and C in, one has this truth table for the outputs at C out, A out and B out, and this conforms to the all-optical Fredkin logic gate. So using a Fredkin logic gate, one can then go for higher computing circuits. So this is a schematic of an XOR and XNOR gate, a full adder circuit, a full adder avoiding fan out, a demux multiplexer. The same configuration, right, can be used by using different select lines. One can activate these switches at different instances, and one can have output coming out of port 0 to port 3. So one can have a demultiplexer or multiplexer configuration. Similarly, a half adder subtractor, a full adder subtractor, or the demultiplexer here using BR coated microcavities, and an implementation of the all-optical arithmetic unit. So the interesting thing is you have optical fibers, they are the backbone of communication technology. You can network data, and you in a computing format, you can use these microresonators along with optical fibers to effect computing. Another interesting thing which I would just delve on is the speed of light has intrigued us for ages. Now the fact is, can you exceed the velocity of light, or can you slow the velocity of light? So the question is, if you have light passing through any medium, we know that it slows down. So that's the definition of the refractive index which characterizes this. The fact is that if you have a pulse of light, you have the group velocity. And this group velocity can be dramatically reduced by two ways, and that is electromagnetically induced transparency and coherent population oscillations. So people started first using low temperatures, so high density materials, Bose-Einstein condensate, rubidium atoms at 360 Kelvin. So they reported a group velocity of 17 meters per second, 90 meters per second, but the challenge was to have it at room temperature, and at room temperature in Ruby it was reported to be 57.5 meters per second, 8 meters per second, and finally in bacterial rhodopsin film, using the principle of coherent population oscillations in 2005, they reported a reduction to as low as 0.091 millimeters per second. So that is ultra slow, snail pace light using just bacterial rhodopsin. So we've done a theoretical analysis, again a rigorous one, and that again of this using the experimental results reported, and you find that if you use a blue light, you can control the delay of the pulses. So theoretically also we've been able to get good agreement with the experimental results, and of course one can optimize these for different pump intensities. So to conclude, there are optimized conditions which one finds when one looks into the application of photochromic proteins for these kind of applications. So you have to look into the factors like concentration, pulse width, and absorption cross sections. You can control the switching characteristics by controlling the probe wavelength, the rate constants, probe intensity. You can see all these factors which can be controlled: the profile, phase shift, time, contrast, group velocity. There are trade-offs between switching time and power. So if you are working at very low powers, right, ultra low, then of course you have to trade off with the switching time, and you have very high contrast depending upon the spectral overlap, and of course you can implement ultra fast operation by looking at the early intermediate or suitable mutants and variants that have a truncated photocycle. Now, of course, there are several aspects for further investigation. That is, bacterial rhodopsin is a unique case. It's extremely photostable, but what about the photostability of plant photoreceptors? Tailoring the spectral and kinetic properties to suit device applications, and how would one operate devices at the single molecular level, right? So that is again a challenge which we've still not understood. How does the signal transduction takes place in a plant or a plant system? So as light comes in, how does that trigger its response and therefore makes it perform a particular function? And of course, if it's a single molecular level, then how would one look into the cascading of these responses or devices? And of course, one would have to investigate new architectures for applications such as parallel computing. So there are several advantages which we come across after this study. So you have low power operation, fast operation, simple design, flexibility by tailoring the properties. You have a multifunctional response. The same material is photoelectric, photochromic, and it acts like a proton pump. So one can implement multiple devices using the same material. Multi-wavelength operation, you can control various features, and if you add on it or integrate it with a system as versatile like a micro resonator, then one can really have a tremendous control over various features, and of course it could lead to a cost-effective proposition and probably lead to a prospective technology. So I would like to acknowledge my PhD students, MPhil, MSc students who've done work on different aspects which I have presented. Our collaborators, Frank and Topolancic at Harvard University, Professor Kamo and Kikukawa, who discovered the phenomenon Photoreceptors at Pioneer University in Japan. Professor Tokunaga, again a pioneer in photoactive yellow protein. Professor Deepak Mathur and A. Dharmadhikari at KFR Mumbai. Professor KPJ Reddy at IC Bangalore, and of course the funding agencies. Thank you very much.